Good day, Homo sapiens. So, today our chapter is prokaryotic cell structure and function. This is the third and new chapter, and today we will be discussing about the general shape, size, and arrangement of the prokaryotic cell structure. We all know that prokaryotic cells are a different type of cells from the eukaryotic cell, but still high in number, and are very important in studying and understanding even the eukaryotic structure and the molecular biology or the molecular levels of study in biochemistry or anything. All started from studying the prokaryotic cells. If we understand the prokaryotic cells machinery and working, it becomes easier for us to study even the higher organization cells. So in this chapter, the shape, arrangement, and size. What do they mean by this? The bacteria. Oh, all right. So the prokaryotic cells that we are talking about are only the bacteria and the archaea. There are two types of prokaryotic cells. Even though people are mostly familiar with just the bacteria, but archaea are an important classification in this kingdom as well, in this domain. The bacteria and archaea are different from one another through certain characteristics, and the one that we know are only that the ribosomal RNA are different from one another. They live in the extreme conditions, the archaea, while the bacteria live in moderate environments, right? So the structure of the archaea or the biochemical composition of the archaea is not studied very much, unlike bacteria. So that's why the organelles or the structures that we are going to study in this chapter will be the general one within the bacteria and the archaea. But today, the shape and arrangement and size, we are only focusing on the general appearance, not the structures, not the internal organelles or anything. We are just studying the outer appearance, the morphology of these bacteria. By the way, the other organelles include the ribosomes specifically, because ribosomes works, I think, 70% of the bacteria, or maybe more, or maybe a little bit less. There are vesicles, they are a lot. Inclusion bodies are a lot of in a lot of quantity present in the bacteria, and there are nucleoid, of course, present. So we will deal with these later. Our cell membrane or cell wall are flagella, or you can say endospores. So in shape, arrangement, and sizes, coming towards the shapes, there are four main shapes of the bacteria, or you can say bacteria, not the prokaryotic cells. The four main ones are the cocci, spherical shape, the bacilli, the rod shape, and the vibrio, the coma shape, which is mostly similar to the bacteria, but they live singularly and in a coma shape more likely. And there is another one, the spirillum, the spirochetes, you can call them. They are in spiral shape. As you can see, they are also resembling to the bacteria, and there are other complex type of structures in the bacteria as well, and I think that is related to these vibrio, specifically because they are quite flexible. Even though we generally talk about the cocci and the bacilli shapes, the spherical and the rod shape, these two are not that uncommon as well. They are present in our surrounding. Coming towards the size of these bacteria, so they range in micrometer range, mostly micrometer range. There are larger bacteria and the smaller bacteria. The larger one, you can say, they found till now is the where was it written? Margarita or something like that. I completely forgot the name. Yeah, Epulophysium, Fischelsoni. Well, there is another one, then margarita one, that is even more larger than that. This one size range from 89 micrometer to 600, like they are very large. While the smaller one you can call the mycoplasma one ranges from ranges to 0.3 micrometer, right? So we think that the bacteria, all that we have, is ranging in the micrometer, right? There are bacteria that are even smaller than the current one and even smaller than the 0.3 micrometer, even though they are rare. We call them nanobacteria because the scale goes into the nano. So mostly we have in micrometers, but some we have in nanometer as well, even though the scientists thought that the bacterial machinery don't allow the bacteria to become even more smaller than this, or in case of that epithelium as well, they thought that it couldn't be larger than a certain size, but their environment they exist. Right, environment is surprising. If you see at this, the oscillatoria, this is big because, but it is not as big as that margarita and the ribofficium. It is, if you compare it to how large it is approximately around red blood cells, right? The E. coli is average. It is not large and it is not smaller. And if you look at the pox virus, it is of almost of the same size as mycoplasma, the one that we studied, that is smaller. So we will be coming towards that. I'm just giving a general review. Now, if you come towards the organization, that the cells are organized in which way, or what is present inside their cells. Are all of them the same inside and out? No, that is not the case. Even among the prokaryotes, some have different bodies than the other. And even among the difference between the prokaryotes and the eukaryotes, the organelles are not there. Scientists thought that if you become prokaryote from eukaryote, right? The only, or not scientists but people thought, only the organelle numbers will reduce, right? But no, even though the organelle number reduce, there are some organelles that are only specific to the prokaryotes and not the eukaryotes. And even in the prokaryotes, there are some prokaryotes that they have these organelles in them, and some don't have, and some have combination of them having and not having them. And other than that, the chemical structures, well, plasma membranes and the cell walls, I think they are not that much different between prokaryotic cells, but the internal organelles number, their size, and their characteristic changes with each prokaryote. So, and I think in the last we are going to study this table, or you can say box, the monstrous microbes. They call it monstrous because of their size, because they thought that the prokaryotes couldn't get bigger than this, and here they become even more larger. I haven't read it, so from here I think, so I don't know if there is anything other than what I have said, or if my point of view is different. See, they are calling it giant bacteria. Like I said, the bacteria are usually average in size in micrometer. These are just the exceptions, like exceptions are present in everything. So let's read. It's not that difficult. I'm not going to read it completely. I'm just going to, go through it. Wait, why is not moving to that side? Yeah. So this is the line of the day, that one might expect that small, relatively simple organisms like prokaryotes would be uniform in shape and size, right? We would think that they wouldn't vary that much because they are simple, but no. They are simpler from eukaryotic cells, but they are not simple in their own right. If you go only towards them, this is not the case, as the microbial world offers almost endless variety in terms of morphology. 3.1 to 3.2 figures. I think we will go towards the figure after we study the structures, right? However, most commonly encountered prokaryotes have one of two shapes. Cocci, coccus are roughly spherical cells. They can exist as individual cells but also are associated in characteristic arrangements. They are frequently useful in their identification. Alright, I don't want to read all of this. I will just say that the cocci are the spherical shape, and they exist in chain form or in pair form or in cluster form. There are three forms of cocci that they exist in: the pair form, the chain form in one plane, and the cluster form. Also, they exist as singular cocci, or you can call it coccus, the singular. Now, the one that exists in pair are known as diplococci. The one that lives in chains are known as strepto, I guess, or staphylo. Let's think if I'm not making any mistake. Yeah, strepto. The ones that are in chains, they are known to be strepto, right? While the ones that are in clusters are known as staphylo. Now, there is a complexity in the cluster formation, right? When you make a cluster of these coccus, they are made in different planes. They are made in 2 planes or 3 planes, like if you look at from X, Y, and Z. Usually they only form at X, right, if it is in chain form, but when it is in cluster form, they are in different planes. Now, the thing is that if you cut these planes, do they all divide the same way? The answer is it depends on the bacteria, that how many cuts it will take for them to be equal. Like for some bacteria, if you cut them or divide them, you can say once it will look all the same. Like for example, in case of tetrads means 4 cocci. Why is it not moving? Hey, move. See the symmetrical cluster. Now, they can be in tetrads or in cubical forms, or cubical packets they call them. Now, why did they call this 8? If you look at it from the 3-dimensional, don't think it like this. This is 0, this is 0, and this is 0. No, make another 0 behind that and it will become a 4 cubical packet, right? Just the shape won't be the tetrad, or you can say square, but it will be a cubical shape from wherever angle you look at it. Now the other shape, well, look at the cocci shape. If you look at this, which form do you think this is? Streptococcus aureus or Staphylococcus aureus. Which one do you think it is? I think aureus is both in cluster form and the chain form, but this one specifically refers to the cluster one. Look at the clusters that are formed. Well, if you look at this one, this is in chain form. It is quite visible. Like if you look at this one and ask from yourself, what is this? Is this chain? It apparently has a spherical structure, so of course it is going to be a cocci bacteria, but what kind of cocci? Now coming towards the bacillus, right? Rod shape. They are commonly known as rod rather than bacilli, because there are some bacteria that are known as bacilli, but they are rod shape. Now bacilli also, like coccus, make chains, although they don't make clusters, the bacilli one. The bacilli has a different characteristic, and that is that their ends, if you make a rectangle and take their corners, right, the bacilli has the characteristics that it has different ends. If it may be more rounded, it may be more pointy, or by the way, the cocci, the spherical shape is rough, and the bacterial of course, rod shape is also rough. It is not completely straight lines or completely circle, right? Now the cocobacilli. Now there is another type that is the combination of your cocci bacteria and bacilli bacteria, only in shape. I'm not talking about the characteristics. That is because the mid section looks like a bacilli and the end section looks like, right, coccus. So you can differentiate if it is a cocci or if it is a bacilli. It resembles both. And they are of course short in size. Now the bacilli, the end shape may vary and can be flat, can be rounded. Flat means it will be pointy, rounded means of course it will be rounded. Cigar shape, it will be kind of cylindrical ends, right? Cylindrical. And bifurcated means fork. Forks will be formed if 2 or 3 bacteria combine. Even if they don't form chains, of course they will be forming chains, but their forms will be chained in a fork form. If they made one fork, then the end of that fork will make another fork of 2, and this end will make a fork of another 2. When the 2 others are formed, they will make further 2 and 2, then 2, 2, 2, 2. All of the ends make 2, attach 2 more bacilli to them, and that's why they are known as bifurcated. Although bacilli or roll shape occurs usually singularly, right, in single form, but some forms in pairs or chains. Of course, the pair form is also present in bacilli. There is just not clusters in the bacilli. Now there are other 2 shapes, right, which are not as common as the bacilli or coccus, but they are not uncommon as well. The vibrio shape, right, comma shape. Sorry, I thought I said that the vibrios are flexible, right? Or did I say spirochetes are flexible? If I said it wrong, then let's correct it. The spirochetes are the flexible ones, not the vibrio ones. Yeah, I think I said that the vibrio ones are flexible. Of course, vibrios are not flexible, they are just like bacillus and just a little bit curved. Although vibrio has shapes as well. Like look at this one. This is a vibrio bacteria. Look at this bud formation and look at this bud formation. It has a complex form. Or maybe this one is also spiralum. Anyway, the spirochetes are in the spiral shape. If you look at this okay, the spiralum bacteria or the spirochetes are actually longer in size than the bacilli. That's how they differentiated. And unlike bacteria, they are not that straight, like there are curves in their structure. That's why they are considered spirochetes. Like when I was looking at this diagram, I couldn't see like where is the spiral. Like we have seen springs and that has a lot of curves. But if you look at this bacteria, you don't see any curves, that much curves. But if you look at this one, this bacteria, it has curves, not that much, but you can clearly. Some bacteria don't have that much visible curves and some have visible curves like this one. Look at this one as well. Even though they don't even seem like bacteria, but this is the cluster formation of the bacteria in a kind of connection or system, which we call the hyphae, mycelium, and that's only present in the actinomycetes. The actinomycetes resembles both bacteria, has the characteristics of bacteria and the filamentous fungi. So spirochetes are curved, more flexible, and have unique appearance. There are, even among the spirochetes, there are shapes that are quite unique. For example, actinomycetes form mycelium and their bacterial shapes look like hyphae of the bacteria, even though they are not. While some look at this, some has these bird-like characteristics or shapes and bulb-like characteristics as well, and which forms hyphae as well. Oval to pear shaped, produces a bug at the end of long hyphae. They do call it hypha, but it isn't hyphae. Remember that. Other bacteria, because hyphae is completely different thing. Other bacteria, that's not that different. All are made from organic compounds. Other bacteria such as Gallionella produce non-living stalks. non-living stuff, a bacterium that has the characteristics to produce non-living characters like plants. When the bark is dead, right? And the bark is produced by the living cells. I think bacteria do produce stalks like that. And few prokaryotes are flat. Flat means flat because their thickness is reduced to almost invisible, or you can say uncountable. And they are talking about the flat-shaped bacteria that they ranges from 2 to 4 micrometer in length and only 0.25 micrometer in thickness. Look at the comparison. Where's the 4 micrometer? Where's the 0.5? It's considerably low number for thickness, and especially for prokaryotes, which are even smaller in size. Now, the prokaryotes also have variable in shape, like some prokaryotes, even if you look at the species, even the species will be present in different forms. Like, so you can't decide if you are going to put them under the category of bacillus or if you put them under the category of coccus. Like, you can't decide because one scientist found it in a bacillus shape and one found it the same characteristic, the same everything in another shape. So you call that pleomorphic. Morph means form, pleo means different. And they gave the example of Corian bacterium. They are saying although they have generally roll-like forms, but we can decide because it can exist in different forms. Not the same bacteria, but the bacteria of that same species, right? Now, talking about the shape, I don't think we have to go it in a detail separately. Now, they are starting from the Escherichia coli. Why are they starting from the Escherichia coli? Because Escherichia coli is very important scientifically, right? They are the bacteria that are studied the most. They are the bacteria that are cultured the most because they are easy to handle. They are easy to find in the environment. They are even present in our own body. So they always start from that because the microbiologist, the first example I think you get is the Escherichia coli. So they are average size, like from 1.1 to 1.5 micrometer wide to, I mean, it is on the smaller spectrum of the bacteria, not on the larger, but not on that much in the middle area, but they are almost average. The mycoplasma, the specific type of a bacteria which lacks cell wall. All other types except mycoplasma have cell wall. Mycoplasma for some reason don't have cell wall. Now the bacteria also have another characteristic, and that is the capsule formation, right? So I don't think they talked about here, but cell capsules also add to their size. Now mycoplasma remained the smallest bacteria for quite a long time, but after the discovery they found even smaller bacteria than the mycoplasma, and that was the nanobacteria, and ranging in nanometer, that is 0.2 micrometer and 0.05 micrometer. Guys, even though this is micrometer, don't let it deceive you, because this is 0.2 micrometer. You can convert it into a nanometer range, right? If you consider 1 a micrometer, right? 1 a micrometer, right? If you go back from that, right? Of course, it is going to go in a nano range to make it easier for you, right? Now they are saying even though we have discovered the nanobacteria, we haven't cultured a lot of them, right? Even though they appear to be bacteria-like organisms. Now that was surprising for them because they did calculation and they thought that it was not possible for, or you can say it is not usual, like it was almost not possible for the bacteria to be in that small shape. Now at the other end of the continent, now they are talking about the larger size bacteria. Now the spirochetes are actually quite large. As we have seen in the diagram, the spirochetes bacteria are generally longer because they have They have to make spirals in their body. Of course, they need some length. Well, that is not the logic, but I'm saying. So 500 micrometer in length, while the photosynthetic bacteria oscillatoria 7 micrometer in diameter, approximately to our red blood cells. But this is not the end. A huge bacterium lives in the intestine of a fish, brown sturgeon fish, Acanthurus nigroficus, Apulopicium. When will I get used to this name on my mouth? Apulopicium fischelsoni grows as large as 600 to 80 micrometer by 80 micrometer, little smaller than the printed hyphen. And the other larger bacterium is Thiomargarita nemibensis, I think. And this was discovered in the ocean sediment. I don't know when they reached the ocean bottom sediments. Oh my god. So there are only few bacteria that are larger than the average eukaryotic cells. They are saying that the scientists, or you can say people, usually consider the difference of the prokaryote and eukaryotic cell in their size, right? But they are telling us that there are prokaryotes that are larger than the eukaryotic cells, so don't go for that. And yeah, I think they didn't say anything else. They said that the organelles we will be discussing in the detail in the further chapter, internal membrane bounded, interior appearance, genetic material, nucleoid, discrete region, or whatever. Now these monstrous microbes, I am thinking if I should read it, because I think the video will get way longer. Still, let me try if I can go through it. They are talking about the biologist. I'm not sure if the claim is correct. Do biologists think that, or they distinguish because of the cell size? I never considered the difference of the prokaryote and eukaryote on the basis of their size. I always thought about of their genetic material, right? And the lack of certain organelles. Like, there is no way a prokaryotic cell will have a nucleus. There's just no way. That's just not the category of them having a nucleus. They are always nucleoid. So they are talking about the monstrous microbes, and that is only the Apulopicium. Again, see, my mouth can't read this word. Fischelsoni, and at first they thought that this was a protist in 1985. But then after they did the ribosomal RNA sequencing, they found out that this was a gram-positive bacteria, genus related to the Clostridium. So they studied about this E. fischelsoni and quite some characteristics of them. And this is the size 200 to 500 micrometer in size. And they are saying that despite its huge size, they are quite motile. They move twice their body length in a second, and that's approximately 2.4 centimeter per minute. Well, slow if you look at from the human perspective, but of course it is fast if you look at it from a bacterial perspective. Moving 2.4 centimeter in a minute is quite a feat. Just think about it. Give him 30 minutes and he will go to, I think, 60 centimeter or somewhere. And of course the motility is gained by the flagella. Now the cytoplasm also have a large nuclei. The more bigger you go, the more complex you go, the more nucleotide material will you have, the genetic material you will have. Of course, many ribosomes. The more genetic material you will have, the more ribosomes you will have, because you will need to function that DNA, of course, or RNA. Now they are saying that why is this astonishing, this Epulopicium? Oh my god, Epulopicium. Why is this astonishing? It's because it is unusual in its size, and this is not a small feat, that it is not small or average in size. To become larger, you would need that machinery for you to work. And if you need more machinery, that will become complex and might lead you towards the eukaryotic. But this guy, the Epulopicium, the bacteria, remained as prokaryote even though its size enlarged. Now, they are saying that the Epulopicium. Oh, now I get it. This is Epulopicium. Fish Epulo, I don't know what Epulo. So they are saying that they transfer between hosts because of the fecal contamination of fish food. And how do you get rid of this Epulopicium from fishes? You just starve them. Starve them for a few days and everything is done. Because poor fishes might not die in few days. In 1997, now, up till this time, Epulopicium was the largest bacteria, right? Bacteria. But in 1997, this heady shoals discovered the Thiomargarita nemibens, and that is a spherical bacterium between 100 to 750 micrometer. If you look at the highest range, it is larger, from 600 micrometer, that is of Epulopicium in diameter. And they are talking about 100 times larger in volume than E. Fischelsoni. 100 times. Of course 100 times. I thought it was 100 million times. Because somewhere here they compared them to the E. coli. Of course it is quite larger than the E. coli. That poor thing is in 1 micrometer, 0. micrometers. Oh no, sorry, 1.1 or somehow. Well, yeah. Now, this vacuole occupies 98% of the cell and contains fluid rich in nitrate. It is surrounded by 0.5 to 2.0 microliter of the cytoplasm filled with the sulfur granules. So what? Its size is because of its a vacuole. I thought it would have a complex. Well, whatever. It just have the vacuole. It has a big fridge than its house. Now, the cytoplasmic layer is same thickness as most bacteria and sufficiently thin for adequate diffusion rates. They are saying that the cytoplasmic layer is the same thickness as most bacteria, right? Like the thickness or viscosity is not different, right? The thickness is not about the size, right? It's about the fluidity. And they use the sulfur as an energy. No wonder they have that much storage for the sulfur. And how do they use the sulfur? Nitrate they use nitrate as well. They use them as an electron acceptor for electrons released when, sulfur as an energy source and nitrate as an electron acceptor. Think about our own electron transport chains. Hydrogen is released, right? Because we take electron from that, right? And that same hydrogen goes towards the oxygen. Now, so as in our body has electron acceptor, and I think in our body the electron acceptor is ATP because it produces ATP, the energy, right? ATPase, sorry. Electron acceptor is ATPase in our body, and it produces ATP, and in this case the nitrate acts as that. While the electrons they gained from, as we snatch the electrons from the hydrogen, they snatch it from the sulfur. Now, the discovery of these prokaryotes greatly weakens the distinction between prokaryote and eukaryotes. Now, they are worried. What actually is the difference between prokaryotes and eukaryotes if they can come in these larger size? If you talk about the cell size, of course, not the other characteristics. They are certainly larger than normal eukaryotic cells. Normal eukaryotic cell, guys, this is taking their size larger than the average of the eukaryotic cells. Now they are saying that the size differentiation has been weakened when we study on their size comparison. The best example is Nanochlorem eukaryotum. Nanochlorem is only about 1 to 2 micrometer in diameter, yet it is truly eukaryotic and has a nucleus and a chloroplast and a mitochondria. They are saying, look, Our 1 to 2 microrange has E. coli, while there has the nanochlorm karyotum. Look at just the difference. One is a prokaryote and one is a eukaryote. So of course the size doesn't decide if it is a prokaryote and eukaryote. So they are saying our understanding of the factors limiting the prokaryotic size must be re-evaluated. Of course that this book is not the updated version, so yeah. Now whose diagram is this? The A one. I didn't see A anywhere. Or was I dumb or something? The photograph taken with a pseudo dark field illumination shows a profile. Oh, okay. The first one is Epulopium, while the below one is Ithio margarita. What? Wait. Which one's which? External mucous sheet and sulfur globule? Are you telling me? Then where is this genetic material? Or are these smaller quantities are the sulfur globules? And the E. coli size, where is its flagella that lets it move faster, 2 point something in a centimeter in a minute? Should I waste my time on it? Mucus sheet, internal sulfur globules. Guys, I'm more worried about genetic material. Where is the genetic material of it? I can see the sulfur globules, I can see the mucus sheet, but I don't see a cell. Or am I dumb? Of course this is just showing the outer, and of course it's not a 3D structure. What am I doing? And of course not every cell has nuclei in the center of the cell. The functions of the prokaryotic structures, right? I think we should study that tomorrow or today. Today is best because it is introduction. So plasma membrane, gas vacuoles. Okay. So these are all the organelles present in a prokaryote, right? So the first is plasma membrane. What is its function? It is selectively permeable, means it allows only certain materials to go in and certain to go out. Mechanical boundary of the cell. Mechanical means the mechanical pressure applied on it or inside. If the water increases, and it won't lyse, like it has the flexibility to retain some kind of pressure or to save cell from some kind of pressure. Nutrient and waste transport. Hey, can you close your door a little bit? Location of many metabolic processes, respiration and photosynthesis. Of course, some bacteria has the metabolic system that is for photosynthesis in their cell membrane and in their cell, So detection of environmental cues for the chemotaxis. There are a lot of receptors that take signals from the environment and inside of the cell as well to maintain that semi-permeability, and of course to produce even more signals or to let the signals from outside to inside for the cell to survive, or from inside to outside to inform the other bacteria in case of biofilm formation, right? Gas vacuoles, buoyancy for floating in the aquatic environment. Okay, I didn't know they had that. Well, it works like gills of the fish. Ribosomes, protein synthesis. That is so simple. Protein synthesis, right? Inclusion bodies. Like these are the storage bodies, right? It can be of anything. Carbon, phosphate and other substances, just as we studied one that is sulfur. Nucleode, localization of genetic material. This is just the locus, the location, wherever the genetic material is present, right? Nucleoid, it doesn't have to be in center. Or maybe it is in the center. Well, we will know afterwards. Periplasmic space. Periplasmic contains hydrolytic enzymes and binding proteins for nutrient processing and uptake. The periplasmic space is this material inside the cytoplasm where everything is present. That is the periplasmic space. The cell wall, of course, this is also for mechanical support and the osmotic pressure. This is way more efficient than the plasma membrane in mechanical stuff, right? Not the chemical stuff, but mechanical stuff. This is less proficient in the right. In the chemical, what was I talking about, man? The battery died. Capsules and slime layers. Capsules are actually pretty common, seen in a bacteria that are actually pathogenic. Well, of course it is seen in the bacteria that are non-pathogenic as well, but the bacteria that has capsule around it and are pathogenic are very hard to treat because they are quite resistant to the host mechanism of immunity. It also causes resistance to phagocytosis, meaning getting eaten by the macrophages or any kind of cells, immune cells in our body. They are resistant to that, and they adherence to surface. Are they saying that they are resistant? No, of course they do adhere to surface. But when capsules make very less receptor for it to be out to adhere to surface. I think it reduces the ability of adherence. Or are they talking about the glue-like things? Fimbriae and pili attached to the surface and bacterial mating. Pili. There is a certain type of pili, sex pili, F pili. And that is only for the sexual reproduction of the bacteria. Well, sexual reproduction of the bacteria are not like the eukaryotic system, right? They are quite simple, more like the asexual, but they call it sexual because of the genetic transfer. And the flagella, of course, specifically for movement. Endospore, this is the survival mechanism. We all will discuss these in detail. And yes, this is it. We are done with the introduction. And we will study in detail the cell membrane, each and everything in the cell membrane.